Hello everyone. So today's topic is enzyme. Okay, all about enzyme. So this is the very important part actually. We do need to understand the enzyme in all aspects because enzyme is very essential in our body system, right? It has role in pathogenesis means many disease occurs due to enzymatic problem means enzymatic malfunctioning. Maybe it may be non-functioning, it may be less functioning, or it may be totally not functioning. Okay, so another thing is that so many roles are there of enzyme in the digestive system. In case of digestion, so many enzymes are involved to digest the food whatever we eat. Okay, not only that, even in the biosystematic way means in our body system so many reactions are occurring, right? Those are named as biochemical reaction. Okay, so chemical reaction means anything that is chemical changes, but when biochemical means inside body or body system. Okay, so whatever the changes occurring, that biochemical process, mostly they are catalyzed by enzymes in some form, many type of enzymes. Okay, so we do need to understand enzyme, and this is very important part in case of in case we consider the NEET exam. Okay, in the NEET exam it is very expected that 2 to 3 questions will be asked from enzyme itself. Okay, so let's go. So enzyme, this is a protein. Okay, this is a type of protein means we know that protein means so many amino acids are polymerized to form protein. Okay, so amino acids first get peptide form, then many peptides get included to form the enzyme. Okay, so enzyme has active site, that site which interacts with the substrate molecule means in the digestion whatever we eat, that is interacted by the with the enzyme. Okay, and get disintegrated or to get broken down. Okay, so these are the enzyme. And so active site I have discussed already. So enzyme, as it is a chemical structure and it is a protein, it is a polymerized form. Okay, so many amino acids are there, peptide bond is there. Peptide bond means CONH bond, CO keto and NH amino group, from amino group NH. So CONH, this is the peptide bond. Okay, I will discuss in later in some other videos about the linkage means the structural properties enzymes like protein structures. Okay, so this is the enzymes. Okay, so chemical reactions, we know that chemical reactions very well, right? Chemical changes from reactant to product means some chemical compounds are interacting with each other, then forming some other compounds by breaking the bond and making another bond or another type of bonds. Okay, so this is the reactions. So chemical reactions usually catalyzed by inside the body. This is the enzyme. Okay, so enzyme kinetics. So enzyme kinetics, one thing you need to understand that enzyme kinetics means enzyme is working or acting on the substrate molecules. Okay, substrate is getting converted to product. Okay, so this conversion means breaking of some bonds and making some other bonds. Okay, so this bond breaking and bond making, the rate of this disintegration means the rate of breaking down of bonds and the forming of bonds. This is the kinetics involved means in each second or in each minute how many substrates or reactant is getting converted to product or form, that is the kinetics. Okay, so see one thing here on example, glucose to pyruvic acid means glycolysis. Okay, glycolysis means glucose, that is the source of energy, we know, means the food that is mostly used to get get the energy inside our body, that is the glucose. Okay, so glucose get converted into pyruvic acid. So this conversion involves so many type of enzymes. Okay, so I have discussed already. So enzyme kinetics, how the reactions occurs. Means substrate is that compound on which enzyme works. Okay, so substrate get converted to product. So okay, here this figure will show the this one is very helpful. So you can see this is the diagram. Actually this is the diagram showing the chemical kinetics, the kinetics in behind the enzyme mechanism. So okay, so see, in the x-axis they plotted progress of reaction, means reaction how much reaction already occurred. Okay, so progress, as the reaction progresses further, then that means product is formed more and more. Okay, and in y-axis it is potential energy. Okay, so potential energy means the substrate potential energy. That substrate has a specific energy, means substrate has specific energy. What type of energy it could be? That means translational energy inside substrate molecule. Means this is the chemical compound. So chemical compound means molecules, molecule means atoms. So many atoms are there, so many ions are there, maybe in the ion form or atom form. Okay, so what happens, those inside internal energy, that internal energy. Okay, so that internal that every compound, every substrate molecule has a specific internal energy. That energy actually known as the potential energy, means overall total energy of the substrate. This is the ground state. Okay, so what is happening, what happens when reactions occurs, then substrate is not able to convert into product form in that energy level. Means substrate must have to go across higher energy. Okay, then only it get converted, then only it will be able to get converted into product. Okay, so you can see here what happens, substrate energy transition state. You can see transition state, in transition state substrate has more energy than the ground state. Okay, that potential energy means initial energy. So now this has more energy. This more energy, how much energy we need to convert substrate into product, that is the main thing in enzyme kinetics. Means what is happening, substrate to convert the substrate into product, we need some energy supply. That energy supply is changeable. When we have enzyme, then the energy needed for the conversion of substrate into product is less. You can see from the graph. Okay, but in the absence of enzyme, to convert substrate into product, we need much more energy to supply to the substrate. This is the difference when we have enzyme and we don't have enzyme. Okay, so you can see transition state, activation energy. So activation energy is nothing but the highest energy level of the substrate minus the energy level of the ground substrate in the ground state. Okay, so this difference is higher when without enzyme. So in the transition state you can see much more energy without enzyme. So with enzyme this is less. Okay, so ultimately substrate is converted to product. So this is the basic role of enzyme. That means it lowers the activation energy. It means it lowers the need energy to supply to the substrate to convert it into product. Okay, so okay, substrate to convert. Okay, next. So you can see here nature of enzyme action. So what happens, enzyme first interacts with the substrate. So enzyme substrate complex is formed. Means transition state means in some type of bond, maybe little small bonds means less strength bond, like pi bond, hydrogen bond. These type of bonds are formed in between enzyme and substrate to interact for interaction purpose. And then this bond in the substrate molecule, some bonds are broken down to get it converted into product. So enzyme substrate complex, then enzyme product. Okay, transition state. Still transition state. After that, enzyme plus product is formed. Okay, so obviously I have discussed already active site. Then that site only, enzyme and substrate interacts with each other. Okay. Okay, another thing, when enzyme interacts with the substrate, enzyme, the molecular structure of the enzyme is not changed. Okay, means it has the potential again to interact with another substrate and convert it into product. Okay, so that's why enzyme not changed. So these are the factors affecting enzyme kind activity. So enzyme is a protein structure. This means chemical structure. We know peptide bond is present inside the enzyme structure, means CONH peptide bond. Okay, so what happens, pH, the pH, the level of optimum pH is there, means when we increase the pH, it happens that the enzyme activity also increase, but up to a limit only. After that, increasing the pH leads to decrease in the activity of enzymes. So energy activity gets decreased when we increase further, when we increase the pH further. Okay, so that is when at hot pH the enzyme activity is highest, is known as optimum pH. Okay, you can see from the graph A very clearly. Okay, and graph B, same pattern, means temperature also helps in increasing the enzyme activity, but up to a limit only. After that, enzyme degradation may occur. Okay, the enzymatic structure may be degraded. That's why the action or activity may also get decreased. Okay, that's why temperature, optimum temperature is also there. Another thing, you can see the last one, the figure. I would like to explain that. So see what they plotted. In the x-axis, substrate concentration. Okay, substrate concentration, and in the y-axis, velocity of reaction, means uh, at what speed, at what rate the enzyme is converting substrate molecule into product molecule. Okay, so that is the rate or velocity of the reaction. So you can see, whenever we increase the substrate concentration, as we increase the substrate concentration, the velocity of the reaction is also getting increased. Means you can imagine a vessel where a specific amount of enzymes is supplied. Okay, so now when we supply more substrate, that is helping the enzymes, or that is useful for enzyme to work more. Okay, so that's why the speed of energy and the speed of conversion or velocity of the reaction is increasing as we increase the substrate concentration, but up to a limit. After that, increasing substrate concentration does not affect the velocity of the reaction. That is the steady state. Okay, so the maximum velocity is the Vmax, obviously, and Vmax by 2, this is the very important. Bmax by 2 means the maximum at what concentration of the substrate the velocity of the reaction is half of the maximum speed. That is known as the Km, Michaelis Menten constant. Km is known as Michaelis Menten constant. I repeat, Michaelis Menten constant is the concentration of the substrate at which the velocity of the reaction is half of the maximum velocity. So you can see here the Michaelis Menten constant. Okay, so you can now imagine that one has Km high and one has less Km. Means high Km means that needed much more substrate concentration to get the highest speed, the half of the highest speed. So that means the affinity towards that enzyme and substrate is less. That's why much more substrate concentration is needed to get the half of the maximum velocity. Okay, this is the output of Michaelis Menten constant Km. Okay. Okay, thank you all.